Uh, I'd like to introduce our first speaker. So Dr. James Kai is a professor of computational systems biology at Texas A&M University, College Station, with a joint appointment between veterinary integrative biosciences and electrical and computer engineering. He's the director of the data science core of the CPRIT Regional Center of Excellence in Cancer Research. Dr. Kai works on interdisciplinary studies involving data science, machine learning, and quantum computing, applying to single-cell biology. He's the author of SCGA Toolbox, a MATLAB toolbox for single-cell gene expression analysis. He develops a quantum circuit model for inferring gene regulatory networks with application to single-cell transcriptomic data, and implemented the model with the MATLAB support package for quantum computing. That's really touching on a lot of bases, and I'm very much looking forward to learning more about it. So with that, James, please take it away.
Thank you, Bob. It's a great pleasure to have these opportunities to meet new and old friends. Let me share my screen here. Okay. So the title of my short presentation will be the Beyond the Average. We are going to use MATLAB developed tools to see what we can do with single-cell data analysis. It's amazing that we are at the intersection of these two major technical revolutions. One is machine learning, and another is single-cell technology. I'm talking about the sequence-based single-cell technology. Before this technology become available, we use so-called bulk RNA sequencing. So in that case, for each tissue, and we can get a single vector of the gene expression of this particular sample. Well, this is a vector, and using this barcode system, and we dissociate the cells into individual cells, then we can get at the end this matrix. This matrix normally have thousands of genes and in thousands of cells, and they read out of their expression in terms of counts of the mRNA molecules. So this matrix is really something data scientists and machine learning people really like to play with. And I put this into a so-called bird-eye view of gene expression landscape. This is, as I said, we have thousands of cells and thousands of genes, so there's only a small corner of that big matrix. And the bar of the indicate the expression level of that gene in particular cell. So to analyze this data, we can go either by cell-wise or by gene-wise. If you look at the genes, you can see this gene expressed variably across the cells. And if you look at the cells, each cell will be uniquely featured by many genes expression. So if you have two samples, then for example, we have one sample from a knockout system and another from the wild type before a particular gene has been knocked out. So you use the single-cell RNA sequencing data technology, you can get two matrices. Okay, with two matrices, we have even more approaches can analyze them. But most current analysis is based on the conventional differential expression analysis. This analysis is average or mean-centric, and people only consider the average expression of this particular gene in one sample indicated as blue, and another compared to its expression in another sample indicated in this orange or red color. So this average-centered approach tend to fail, and I will use short examples to indicate why this is the case. This can be extended to a whole lecture anyway. So I'm going to show you, even you don't compare, just use a single sample experiment. You can get one matrix from that particular sample, you can learn a lot about the function of the genes in this given population of cells. And then if you have two samples, instead of using conventional DE analysis to compare the mean expression difference between genes, we should use gene regulatory-based comparison. So I will show some case of this either case. So as I said, if you only use one cell population, say in this case from a wild type, you get a matrix like this. And this is called a single sample experiment. And with this matrix, that's a real data set I got. You see, it's very sparse. There are some genes have a lot of zeros across this individual cells, and the columns are the cells, and the rows are the genes. So what we can do from this population of cells. By the way, there's a same cell type and the same cell states. There's no contrast between one cell and with another. It's a homogeneous population. The simplest way we can do the average, okay? So you can compute the average for this gene and put the number in this column and for another. You can also calculate another metric. It's called drop-off rates. Basically, is how many zeros in terms of percentage for this particular gene. And then you put the percentage here. As you can see, these two metrics are not independent. If you have a gene, have a high expression value average, then you should expect a low drop-off rate. And that's true if you plot them out and their theoretical solution and for this, and you can get this beautiful analytical curve. And once you put all the genes in this using scatter plot on this scatter plot, you see the most of genes follow this regression curve nicely. That indicate all the mean and their drop-off rates are expected. And however, what we are really interested in are those so-called highly variable genes, or are those genes which are standing out from the regression curve. The far away from the curve, the more variable, and they express in terms of this unexpected deviation from the mean. And for the system you studied, you can look at what are those genes, and by the name you can figure out their function individually, or you can group those gene set from the highly variable gene set to understand as a group of function their function, and then to reflect what the population of cell are doing. So this is a very effective method, and we further developed a new method using adding one more variable, which we call the coefficient of variance. So make it a 3D and plot to make this identification of highly variable gene even more accurate. And in MATLAB, we can do this easily, like 3D, and to look at the deviation of the genes from the curve to identify the highly variable genes to describe the function of the cell population. Okay, all the tools and methods I developed have been already put into single-cell gene expression analysis toolbox, as Rob mentioned, and this is a link, and we can find all the information. I will just quickly show you some screenshot, and nowadays you can even open this toolbox using the tools through the MATLAB online, and you get this nice interface, and you play with your data points. This is very important for biologists if they don't have programs in their labs to handle the complex data sets. For example, if you want to download and repeat some analysis from a published data, and you can go geodatabase, get a sample accession number, and load that number into MATLAB toolbox I developed, and then you can get this cell, pre-processed cell, and then with several click, you get this dimension reduction and clustering, and also automatically annotation of the cell type. And this is basically most of the routines have been built in, so you don't rely on any coding, and you can achieve quite a lot. And then you can see I want to extract a particular cell type, and for example, in this case, stem cells, you can extract them out automatically, easily, and to work on exclusively on those particular cell type. And also within this toolbox, we have a suite of high machine learning methods. It's based on tensor and manifold. So this is called single cell 24 suites. And so the first one is called single cell 24 net, is used to compare two networks. You build gene regulatory networks from the two samples and to compare them. And using that, we can identify differentially regulated gene modules. And this, for example, in the traditional DE analysis, you won't be able to find anything difference, but you can detect the subtle change in the network level. Another tool is called single cell 24 KNK. This is a virtual knockout tool. You can simulate once a gene has been knocked out from the system, what will happen. We can use this computational tool to find if you knock out this TRIM2 genes, for example, which other genes will be perturbed. And using this, we can prioritize the potential or predict the potential phenotype and to replace the real gene knockout animal research. We also have tool to study cell-cell interaction. So for giving the time, I won't be able to go details. As Rob mentioned, we also developed this quantum-based gene regulatory network construction model. So using quantum computing algorithms to infer more accurate gene expression from gene expression data to infer the gene regulatory networks. Okay, thank you. And this is my lab and the findings, and we are open to academic and also commercial collaborations. Thank you very much.
James, thank you very much. That was super interesting. I think it's always super impressive to see people gathering like different kinds of algorithmic capabilities and methodologies from multiple disciplines and trying to synthesize them. And so I think your work is a really stellar example of that. So thank you for sharing. Does anybody have perhaps one question from the crowd? We might have time for one question. Okay. Then, with that, let's thank James again. Thank you. And we'll move on to our next speaker. So Dr. Courtney C.J. Johnson is a postdoc in the lab of Dr. Hari Shroff at the Howard Hughes Medical Institute Janelia Research Campus, developing rapid and robust methods to advance adaptive optics for fluorescence microscopy, a topic very close to my own heart. CJ earned her chemistry PhD at Duke, developing new 3D live cell fluorescence microscopy methods in the lab of Professor Kevin Welsher. As a founding member of the Welsher Lab, CJ spearheaded the design of 3D Faster, a new method for faster 3D point scan imaging, and led development of the multimodal 3D Trome microscope for visualizing dynamic virus cell interactions in 3D with unprecedented temporal resolution, building these microscopes from an empty optical table to fully automated operation. So with that, I'd love to hand it over to CJ. CJ, please, can you share more about your work?
Can you hear me?
We can hear you.
Awesome. Thank you for the introduction, Rob.
Of
So
course.
today I'm going to talk a little bit more about my 3D tracking work because I think it's a great example of a concept that kind of guided it, which is this idea of code as a force multiplier for fluorescence microscopy. So something that I really realized when I had accomplished this work was that teamwork and automation can be force multipliers. And I know that sounds like a scary military term, but really what it just means is that a force multiplier is a tool that gives one person or team the power of many. And it cuts at the heart of something we are often trying to do as scientists, which is how can I get more done in less time? And so teamwork and automation can go hand in hand, particularly for microscopy, where we're developing these new methods that can often be highly specialized and require experts to use. Instead, this can enable you to employ teamwork in your workflows because you're no longer limited to just specialists who can use the microscope. And so instead, kind of the end goal that I was trying to get at here was what I would call push button microscopy, where you push a button and it just works. You push a button and you get a data acquired, you push a button and your data is processed. And so kind of these fully automated end-to-end workflows. And this also has the effect of enhancing uniformity, particularly on the processing side, because you no longer have people who are doing things just a little bit differently. You get kind of uniform results, no matter who's doing it. And so what this might look like is you set up an experiment, you acquire data, you process data, you visualize data, and then you sort the data. And then while all that's happening, you visualize more data while that's previously setting up. You sort data while you're acquiring previous data. You set up a new experiment while that data is processing, and so forth. And so you end up with these highly parallelized workflows the more you can automate that. And so a good example of where we really needed this kind of high throughput workflow was with 3D trim or 3D tracking and imaging. And so the heart of this work was we wanted to understand how viruses were able to infiltrate and infect cells prior to the moment of first contact. And if you look at the textbook picture of virology, I'm not even going to talk about all of them. I'm just going to talk about the first part, where it just says the virus attaches to the cell and enters, and that's where it starts. But unlike what this picture suggests, the virus doesn't just magically teleport itself to the cell. It has to progress through these highly 3D barriers in the epithelium that are basically your body's host defenses to stop this from happening. And there are a variety of defenses in this highly 3D space. And yet the virus persists. And so if we were able to learn more about how viruses are able to navigate these complex 3D spaces, that would be very helpful because you can't infect a cell that you can't get to in the first place. But it turns out that if we want to track things at the single virus level, this is typically done through single particle tracking methods. And they look a little something like this. And so this is the most kind of simple example I could give, which is wide field. And it really brings home the need for 3D information because these trajectories, particularly the unlabeled ones, are they on the cell? Are they above the cell? Are they inside the cell? If we don't have 3D information, we don't know what's actually happening. The virus could be doing something really meaningful with the cell, or it might not be doing anything at all, and we have no way of telling the difference. But even if we were to use a more method like spinning disk confocal, where you can get 3D information by optical sectioning, you still run into a problem where the more 3D your space is, the lower your temporal resolution, and thus the ability that you can actually extract information from trajectories really goes down rapidly. And so these methods typically are not well suited to tracking in highly 3D spaces because they use cameras. So we wanted to take a different approach to tracking. So the approach we came up with is called 3D trim. And so the way this works is we have cells and we sprinkle in some viruses. And both of these are sitting on a heated sample holder on a piezoelectric stage and microscope objective that are shared by two chromatically separated microscope systems, one that only tracks the virus and one that only images the surrounding cellular area. And so this is actually a very, very different approach to tracking. Because if I just kind of focus in on our tracking system called 3D Smart, 3D Smart is a real-time active feedback single particle tracking system that can produce richly sampled trajectory over a large 3D area. And it's sensitive enough that it can track single molecules for minutes at a time. And so if I look at this trajectory of 100 nanometer microspheres diffusing water, It occurs over a 30 micron range, which is massive compared to most single particle tracking methods, which can get a handful of microns at most. And it doesn't have to sacrifice temporal resolution to do so. In fact, the temporal resolution is even better, which is why the trajectories are so detailed. And I'll show a little bit later how we can use this to extract information. And again, this is very different from normal tracking. There's no camera, there's no image in this case. Instead, the tracking is done using a real-time feedback loop that localizes the particle 50,000 times per second, and then uses a piezo stage to recenter on that particle. But you'll notice there's no image, right? So, and we also need to image with a moving stage. And so the method that I developed to overcome this limitation was called 3D Faster. And I'm actually not really going to go into detail on either of these, but 3D Faster basically uses a 2-photon microscope extended to the third dimension. And there is a way in which it's done that overcomes the limitations so that it can image while the stage is moving without motion blur. So if you want to learn more about either of these methods and how they work, I highly encourage you to read the papers. But instead, I'll just say that they're combined on one platform, and that is how 3D trim works. So basically, you have a trajectory, you have an image, and then you overlay them, and they are completely localized. So I only have time today to show kind of our best application, I guess, which was our ability to track an image in highly 3D tissue. So we wanted to, again, learn more about how viruses are able to move through these complicated tissue environments. And so to do this, we cultured HT29 MTX cells on a porous membrane, and that allowed us to have these cells that grow into layers. And the geometry is a little bit different than what you might be expecting. And so that's why I've chosen to show here for a brief second. So we have an inverted microscope platform. And because this membrane is mounted upside down, the cells are kind of hanging down from the ceiling, like you can see here. So the virus will be kind of swimming from underneath in this case. And so what this looks like is, so we have our trajectory, which is shown in this purple. This is the movement of the virus. And we have the image that's kind of drawing in as it's being scanned. And so the movement of the virus in this system is really interesting for a number of reasons. One is that it's actually highly confined, and you can kind of see this behavior where it's hugging these cells. And yet it's actually moving quite rapidly. As you can see, it just because it's confined doesn't mean it's slow. It has an average of 0.75 microns squared per second. And also it's not binding at any of these points, right? It's making these intermittent contacts, but it's not binding. And so this is a really interesting behavior because that's typically what is most highlighted is productive binding. But actually, we see a lot of non-productive contact as it interfaces with these cells. And so you can kind of get a sense for how complicated this environment is. And the virus in this case was traveling over a 10 micron range. And yet it was still making all of this contact because of how dense the tissue is. And here the color coding, the red means that the contacts were within 500 nanometers, which is kind of our contact threshold. We say that anything within this threshold, the virus is actually touching. The orange is basically anywhere within a micron, but more than 500 nanometers. And so there's a very high density where the virus is dwelling in this area, but it's actually moving quite rapidly. And if I wanted to try, and if you want to kind of zoom in and look at what the level of detail in our trajectories is, and what it's good for, is we're actually able to resolve changes in diffusion. So while it's not making productive binding, it is being somewhat hindered by its environment, because you can actually resolve diffusive changes. And we can only do this because of the amount of information that we're getting from our tracking. So if I was to try and do this with a state-of-the-art spinning disk over this range with a very similar diffusion coefficient, even if there were changes, which there wouldn't be in this kind of way we're doing here, you wouldn't be able to resolve them because you have too few samples in your trajectory. And so this is a great point of how your temporal resolution directly translates to the amount of information you can extract. And I should also say that automation was really critical here for us, because we were able to take thousands of trajectories and to see that different behaviors were not, we were very sure of what we had. And one drawback to these active feedback methods is they are truly single particle. You can only track one particle at a time. And so having this high throughput was really critical for us, and it was automated end to end. And so, the acquisition was automated, and you get this nice, data that consists of your tracking data and your imaging data, which are, again, different. And then MATLAB was really our critical hub for kind of processing all this data through and making it so that we could then output a file that could be visualized. And so I like to show this as an example of how automation can really enhance your productivity and the types of things that you're able to accomplish. And again, part of why this ended up as great as it was is because I was able to have a great team. And so I would like to shout them out here, Yushin, Jack, and Jonathan, who were able to use the scope and produce this amazing data. And then my PI, Kevin Welcher at Duke. And maybe next time I will talk more about my adaptive optics work, but I didn't have a chance to go into it today. And thank you for your time.
CJ, thank you very much for sharing. That was a really fascinating review of multiple different steps in the process, like from the equipment through the software, and then through the actual practical application of what you had developed. That's really fascinating. Do we have any questions from the audience? And while one may be coming in, I had one myself. It looks like the tracks of the viral tracks that you were looking at were fairly jagged. Is that a real phenomenon, or is that like a combination of resolution limit, or is that is that the kind of expected semi-Brownian motion that you expect at the cellular level?
Yeah, so that's a great question. So if I kind of go back here and so these are obviously very jagged, and so this is from the temporal resolution being low. The more sampling you have, so really it's a line that's connecting your samples. So the more samples you have, the less you will see those kind of acute angles. And so that's a really great point in that your temporal resolution is information because it's your ability to sample those trajectories, if that makes sense.
Yeah. Okay, that does make a lot of sense. So CJ, thank you again. We'll see you at the discussion at the end. Next up, we have Dr. Hojun Suk, who is a co-founder and CEO of a biotech startup called DX Lab. At DX Lab, Dr. Suk has led the development of the company's core pathogen detection technology and its point-of-care diagnostic platform. He's also leading the development and validation of various diagnostic tests for infectious diseases, one of which has been authorized by the FDA through the emergency use authorization for COVID-19 testing. Before DX Lab, he's worked on developing robotic technologies for automated patch clamp recordings, in vivo, and novel sensory stimulation-based treatment strategies for Alzheimer's, using MATLAB as a critical tool for various systems control, image processing, and signal processing needs. He received his PhD in medical engineering and medical physics from MIT as a Samsung Scholar. Hojun, are you there?
Yes, I'm here. Can you see my screen okay?
We can see your screen and we can hear you.
Great. Sorry, I'm not very familiar with the WebEx platform, and I'm ashamed to say, as you did, that after 3
I
years,
made my own gaffe earlier. Yeah, we're all good here.
Yeah. So let me just maybe turn on my video. That's of interest. But yeah, I just want to confirm. Can you see my full screen? Okay. The presentation.
We can see your full screen,
Great.
And we can see your camera. So
Awesome.
Good luck.
Great, thank you. Yeah, so thank you, Rob, for your introduction. But yeah, as Rob mentioned, my name is Hojun. I am the co-founder and the CEO of a startup called DX Lab. But before starting DX Lab, I was a postdoc in Professor Li Wei Sai's lab in the PCAR Institute for Learning and Memory at MIT. And I'm still affiliated with the lab as a research affiliate. But today I'm going to talk mostly about the work that I have done in Dr. Sai's lab at MIT, utilizing MATLAB as the core tool for EEG signal processing and visualization, which was critical for the work that I have done in the lab and also that is continuing in the lab. So let me try to advance to the next screen. But before I start talking about my work, I think I need to first tell you a little bit about what brain oscillations are, or neural oscillations, some people call it. But as you can see in this video, what brain oscillations they're essentially a rhythmic or repetitive electrical activity of ensemble neurons inside the brain. And they essentially are generated either spontaneously or in response to various stimuli. And these oscillations have been first discovered close to about 100 years ago using EEG. But when we look at these brain oscillations, they can actually be grouped into several groups based on frequency. So as you can see here, the frequencies of these oscillations range anywhere from 0.1 hertz, very slow oscillations, all the way to 100 hertz or higher. And as you can see here, based on the frequency, the major frequency of these oscillations, many previous studies have found that these brain oscillations at specific set of frequency bands are closely related to different activities of the brain or cognitive behaviors. So as you can see here, for example, if you see the delta waves or delta oscillations in your brain, then those are highly correlated and associated with detached state of your mind, awareness, or healing and sleep and so forth. Versus if you go up to the higher range of brain oscillations in the gamma range, those gamma oscillations have been correlated with the higher insight, peak focus, expanded consciousness, and so forth. One thing that our group, as well as many other previous studies, have noticed is the fact that the gamma oscillations, the higher frequency brain oscillations, are reduced in Alzheimer's disease, both in mice and in humans. So on the left here, I'm showing you an example recording of gamma power or gamma oscillations in the hippocampal area called the CA1 in the brain during a quiescent behavior of mice. And as you can see here on the left, in the wild type mice that are healthy, you can see that the gamma power is pretty strong. But then compared to that, on the right, the 5XFAD mice, which are the genetically modified mice for representing Alzheimer's disease in mouse models, these mice show much lower gamma power compared to the healthy wild type mice. In humans, we've also seen, or other people have shown, that the gamma oscillations are reduced in the patients with AD versus cognitively normal subjects. So in this example, on top, we see the brain recordings done using magnetoencephalography or MEG of an elderly cognitively normal subject. And on the bottom, you see the patient with AD. And the dotted lines show the cortical recordings, and solid lines show the more deeper brain region, the thalamic recordings. And as you can see here, the gamma oscillation, especially in the cortical region, is significantly reduced in the patient with AD versus cognitively normal subject. So having seen all of these recordings and studies that show reduced gamma in Alzheimer's disease, our group asked the question, can we actually restore gamma oscillations to mitigate disease pathology in Alzheimer's disease? So to test this idea, we designed 40 hertz sensory stimulation to drive gamma oscillations non-invasively and see if that reduces pathology in mice. So we initially started with the light flicker, which we have and others have noticed that can non-invasively increase gamma oscillations in the brain. And once we did that, we confirmed that the gamma oscillations around 40 hertz was indeed increased utilizing the 40 hertz light flicker in the mouse brain and the visual cortex area, or VC. And we saw, surprisingly, that increasing gamma oscillations in the visual cortex was able to reduce various pathology markers in the same area in the mouse brain, ranging from the amyloid plaques you see on the top to the tau pathology at the bottom. But also we saw the microglia cells, which we call essentially the janitors of the brain, becoming more active, which we hypothesize that they may be playing some role in clearing the amyloid plaques and tau pathology from the brain after being stimulated with our 40 hertz light stimulation. We also tried to look at if we can also induce similar behavior or similar response utilizing a different sensory modality. So we utilize a sound as the sensory stimulation modality, and we were able to show that just like the light, when we flicker the sound at mice, we were able to induce 40 hertz gamma oscillations in the auditory cortex of the mouse brain, and also similarly to the light stimulation. With the sound stimulation, we were able to reduce amyloid plaques and tau pathology in the same area, and also activate microglia in the brain. In addition to that, we saw with the auditory stimulation, we were able to induce glia and other vascular changes that we also hypothesized to be responsible for clearing different plaques and debris from the brain. And also we saw improvement in cognition, especially around spatial and object-related memory in mice that were treated with the auditory stimulation compared to the mice that were not treated with the auditory stimulation. We also went on to combine both the light and sound stimulation at the same time to see what additional effect that may have. And we saw, as you see in this figure, with the combined light and sound stimulation, we were able to not only clear the plaques and different pathology in the respective sensory cortical areas in the brain, but also throughout the entire cortical region, as you can see on the right, with the combined stimulation and also other parts of the brain, we were able to reduce pathology in those areas. So seeing these effects of sensory stimulation, non-invasive sensory stimulation, we now ask the question, can we actually use sensory stimulation, which we call GNS, can we use that to have impact in humans, and especially patients with Alzheimer's disease? So that's why we went ahead and set up a new device that's designed, optimized for human studies and human use. So as you can see here, we essentially combined a light panel that we programmed to generate a light flicker at the desired frequency that we wanted to generate for visual stimulation. And also we added a sound bar that we were able to control to generate the auditory stimulation for generating gamma oscillations in the brain. With this device, we initially started the characterization of brain oscillations with our sensory stimulation device. And this involved the electroencephalogram, or EEG. With the EEG recording, we essentially wanted to look at, does our stimulation induce the gamma oscillations in the brain that we can look at, or that we can quantify utilizing the power spectral density, and also visualize where in the brain the stimulation is having impact with the oscillations. But to get there, MATLAB was a critical tool that we had to utilize with all the different toolboxes that are available from MATLAB itself, but also from the MATLAB user community. For example, there's a great toolbox called EEG Lab that we have utilized heavily to filter and analyze the raw EEG data that we were gathering. So we were also especially utilizing the signal processing toolbox available from MATLAB to look at all the different frequency responses and other numbers that we wanted to look at to understand the impact of our stimulation on brain oscillations. So utilizing the EEG recordings and also MATLAB toolboxes, we were able to look at the gamma oscillations in response to our non-invasive sensory stimulation. And as you can see here, we started out with testing cognitively normal young people. And in these patients, we were able to see that with our stimulation, just like in mice, we were able to generate gamma oscillations with our sensory stimulation. And we also confirmed that the same thing can happen in the cognitively normal older group, as well as the patients with mild AD. We further characterized the difference between the visual stimulation versus auditory stimulation versus the combined visual plus auditory stimulation. And as you can see in this figure, the right column shows the combined stimulation on each of the age groups that I just talked about. And as you can see, we were able to show that the combined stimulation had the most widespread impact and was able to generate the most widespread gamma oscillations throughout the brain that we were recording with the EEG. So from that finding, we went ahead and also looked at the impact of our sensory stimulation on gamma oscillations in deeper brain regions, because EEG, our non-invasive surface EEG, usually records the data most strongly from the superficial cortical layers. But we wanted to look at the impact of our sensory stimulation in the deeper brain regions. So we recruited patients with intracranial EEG electrodes implanted in their brain. And in these patients, we were able to confirm with our sensory stimulation that deeper brain regions can also be induced to generate gamma oscillations with our non-invasive sensory stimulation. From these findings, we then went ahead and started a small pilot feasibility study where we recruited a handful of control patients as well as active patients, where the control patients were exposed to our sham condition, and then the active patients were exposed to our 40 Hertz sensory stimulation for 3 to 4 months, and they were exposed to our stimulation every day for an hour every day. And we looked at the impact of the repeated 40 Hertz sensory stimulation in these patients with mild Alzheimer's disease. We first looked at their structural changes with our sensory stimulation. And this figure that you see on the left here is an example MRI image of a control patient after 3 months. And the area that's shown in blue is essentially showing the area where the brain has atrophied, meaning you lost the brain cells and the brain has actually shrunk, which is typical in patients with mild Alzheimer's disease in several months. But what we saw was, interestingly, after 3 months of being exposed to our stimulation, the patients who were exposed to our 40 Hertz sensory stimulation did not show much atrophy in their brain compared to the control group, which showed the typical amount of atrophy in their brain. Further, we also saw that the change in the hippocampal volume was different between the patients who were in a control group versus the patients in the active group. Specifically, the patients in the control group showed a significant reduction in their hippocampal volume, which is again typical in patients with mild Alzheimer's disease, but patients in the active group did not show a significant difference in their hippocampal volume. We also looked at the impact of our stimulation on what's called interdaily stability. It's essentially a measure of how regular your rest and activity levels are throughout the day. And this interdaily stability has been studied heavily in the area of Alzheimer's disease and for its potential association with the disease progression. But as you can see here, we were able to show that after several months of our stimulation, compared to the control group, the active group was able to improve their interdaily stability versus the control group, which actually had a worsening interdaily stability over time. Lastly, we also looked at the impact of our stimulation on memory in these patients. And as you can see here, we were able to show that in patients that were in the control group did not show much difference in their memory recall ability versus the active group that received our stimulation for 3 months, which was able to show an improvement in their performance in the associative memory recall activity. So we're very, very excited. Although this is a small study, we were very excited to see some of these effects of our stimulation on patients with mild Alzheimer's disease, and more studies are being designed and being conducted in my old lab to really understand and better characterize the impact of our sensory stimulation on this very difficult disease. So with that, I would like to acknowledge my old PIs and advisors, Dr. Li Wei Sai and Ed Boyden, and also Dr. Emery Brown, who was heavily involved in the project, and everyone else in the human gamma team in Dr. Li Wei Sai's lab, from Diane Chan, Brandon Jackson, and everyone, Noah Milman, who worked really hard to make this work happen, and also who are still continuing this great work, and everyone else who contributed to this work. So with that, thank you, and happy to take any questions.
Hojun, thank you very much. What an amazing tour of different data types and methods that you've used here. It's really, really quite interesting. We do have one question from the crowd, from Valentine. Are the light signals a sine or square profile? And same, is the audio a pure sine sound?
Great question. So both light and sound are square light signal. So sharp rise and I guess fall in the light and sound as well. In mice, however, the sharp rise and I guess fall in the shape of the sound is overlaid with the sinusoidal 10 kilohertz sound as well. So, but in humans, we're utilizing just a square wave, if you will.
All right, then thank you very much. I think we'll move on to the next talk, but Hojun, thank you again. Looking forward to the panel discussion.
Thank you.
All right, Up next, we have Josh Caputo. All right, there you are. Josh is the president and CEO of Humotech, a company that he spun out of Carnegie Mellon. I know how to pronounce that properly. In 2015 to provide the world's first hardware and software platform that supports researchers and developers pursuing innovations in prosthetics and orthotics, exoskeleton technology, and other wearable machines. Trained as a multidisciplinary engineer and scientist concentrated on robotics, his work focuses on addressing gaps in how wearable products are invented, designed, and fit to human users. Josh's driving passion is to help realize a world where everyone can access the devices they need to lead happier, healthier, and more productive lives. Very much looking forward to learning more.
Thanks, Rob.
Right.
Is my audio working? Is my screen share working?
Audio working, screen share working, camera working. Looking
Super.
forward to learning more. Take it away, Josh.
I've successfully figured out WebEx. Thank you. And this will be a very different take on the concept of bioconvergence, more macro and musculoskeletal. So yeah, my focus in my research at Carnegie Mellon and now in my work at Humotech, the company I founded, is to use the idea of emulation applied to wearable machines, which is a word I made up to encompass the types of devices you see here on the slide: exoskeletons, exosuits, prosthetics, wearable robotics, and the like. So the basic challenge that I work to address is that developing, testing, and fitting these devices is incredibly difficult because individuals' outcomes are hard to predict and people are complicated. And this makes it very hard to justify new technologies when bringing them to the clinic, when trying to deploy them into the world. Slowing adoption, slowing the pace of research and innovation. So our mission to transform the way the world develops and uses these devices is to leverage our robotics platform. You're probably familiar with the idea of simulating human walking, running, human mobility in silico. But we find that those approaches are not accurate enough. So what we do is emulate the mechanics. We have a real human walking, running, jumping, and we use off-board computing and actuation to mimic the mechanics of either actual products, real-world products that we've tested, or hypothetical products, new concepts that we're developing. And yeah, we believe the key to kind of unlocking the future that all the science fiction paints for us is human in the loop optimization. And this is sort of a logical extension for me of all the traditional kind of real-time computing hardware in the loop approaches that are commonplace in automotive, aerospace, and whatnot. So I'm speaking to a MATLAB audience, so I don't feel ashamed of showing you a complicated flow chart. This is my depiction of sort of R&D today. So today you start with some mechanical criteria, some idea about what the wearable machine should do, and you embody it in CAD, and you can test that in finite element simulation to determine, whether or not it breaks and whether or not you're meeting your stiffness criteria. You can iterate on this quickly thanks to computer-aided methods, but ultimately, once you pass the tests within the computer, you have to build a physical prototype to test this in vivo on a person and evaluate whether or not the user likes it, whether or not it helps their mobility. And if that's a yes, well, good for you. You can move on to design for production. But more often than not, you realize human idiosyncrasies and challenges you didn't expect, and you have to go back and revise your design criteria. Now, this loop can be really slow. At best, it's days to revise a design and build a new prototype. At worst, it could take months. So our technology is all about improving that the speed of these design cycles. So we enable the testing with the human to happen at the same time as the computer-aided design and finite element analysis. So before you've fully realized the device in a physical prototype, you can evaluate these questions of, is it working for the user subjectively and objectively? With our technology, these cycles can happen in minutes or hours. So definitely a 10x in speed. And then you only bother to physically prototype the devices which meet requirements that have already been very well refined. All right, so what the heck does this look like? There are many different embodiments of the Humotech technology. I'm going to show a few examples, and then we'll dive deep into one of them. This example is our hip exoskeleton, and it is a bilateral device, so assists both hips in both flexion and extension. And this is tethered to our large multi-axis off-board actuator unit. It can apply a ton of torque and power for high-performance activities. The whole idea behind the off-board actuation is that if you were to take these motors and put them on board the person, the device would be so heavy that it would confound the explorations that you're trying to do. Sorry, it's not advancing. There it goes. But we also have a portable version of this platform with motors, computers, and power that are much smaller and fit into a backpack form factor. Here it is depicted with our bilateral ankle exoskeletons. And then the third example that I'm going to dive into a bit more detail on is our prosthetic version of this system. Focusing on this example because it's kind of where it all began for me back in the lab at the university. And also, this has sort of grown into our largest collaboration with the VA, DoD, using congressionally directed funding. So here we're focused actually on emulating conventional products. So rather than dreaming up sci-fi possibilities, we're very focused on clinically translatable methods. And what patients and clinicians need most today is tools to better select from among the hundreds of options that they have for prosthetics componentry. It's a very wide and overwhelming design space that up until now, clinicians have had to rely on intuition and sort of trial and error, in the field to find what works best for the individual. We can instead rapidly iterate on different design possibilities in the clinic, generating both objective and subjective feedback in real time. So the way we do this is first by mechanically testing an array of different products. These are just some random products that I selected off the internet, not the actual ones we used in the study. But the idea is that the clinician and patient work together to determine a set of potential products. Before this all happens in the clinic, Humotech has tested these products in a machine, or in machina, as I like to say. From that, we generate mechanical response profiles, which we then emulate with our robotic system. So for each one of these products, we determine the relationship between torque and angle about the ankle joint. In this case, we're looking at prosthetic foot and ankle mechanisms. and then we program these into the computer. But remember, you could do the same thing for hip exoskeletons, ankle exoskeletons, whatever, prosthetic arms, whatever you can imagine. Humotech as a company has worked hard to kind of demonstrate multiple modalities of this approach in an attempt to kind of demonstrate its universality. So then in real time, while the person is walking on the treadmill or stair mill or whatever other kind of moving walking surface, parameters can be changed through our GUI. And the response can be read out in real time. Here, we're not showing any sort of objective or subjective outcome measures. This is just sort of very directly the state of the device, the control system controlling the ankle. But you can imagine measuring, the full kinematics of the lower limb. You can measure metabolic rate while the person walks. You could observe electrical activity in their brain. There's really a plethora of ways to analyze gait. And our platform sort of doesn't discriminate. We leave that up to the investigator to decide what's most interesting to them. But in the study that I'm kind of showcasing here, the focus has been more on the subjective outcomes side. So there's a structured survey that was developed at the VA where patients report sort of self-reported measures of preference, of energy exertion, of balance. And the purpose of the study is to test the predictive validity of this emulation paradigm. And these are just some preliminary results from the study showing that both within the lab and after a 2-week take-home trial, the brief experience with the emulator is predictive of people's preference for the actual devices, which is pretty exciting for us. So we are kind of replicating this study in other domains, one of which is ankle foot orthotics. And several more new studies are starting up as well along these lines. Before I wrap up my slides, maybe I should have put this sooner, but the presentation came together at the last minute. Let me talk a little bit about MATLAB and Simulink. So the programming interface for our whole system is based in MATLAB Simulink. And here's just like a very high level snapshot of what that Simulink controller looks like. Our whole purpose as a company is to kind of lower barriers to getting involved in this kind of research and development. Historically, it's really only PhD level roboticists who've really been able to experiment with different device design control strategies. But we've designed in Simulink a controller that's super easy to learn and customize for both novice users and advanced users alike. This really differs from a standard commercial wearable robotics platform, which is generally not designed to be modified for perhaps obvious reasons. But yeah, we love our R&D customers and try to provide the maximum amount of flexibility. And then in parallel, we have the graphical user interface that we developed as a standalone application, which interfaces with MATLAB. The code is deployed and runs on a SpeedGoat real-time controller. So yeah, that's Humotech, and happy to take any questions or talk afterwards. Thanks.
Josh, thank you very much. So I'm curious. Do you see this as extendable to a long-term engagement, like continuous refinement for a very long time, like iterating toward iterating toward an optimum, or is this more meant to be a shorter term solution?
Yeah, that's a really good question. Optimization ideally occurs, in my opinion, on multiple timescales. You have in perhaps the fastest sense optimization or opportunity to optimize on the millisecond timescale. Really ensuring that you're applying the right torque at the right time, mid stride. That obviously requires a robotic system to implement. Most devices out in the real world are not robotic. Most devices, prosthetic and orthotic devices, are simple fiberglass or carbon fiber springs. So while we're very excited about that possibility of optimizing on fast timescales, it's, in our opinion, not super practical right now because there just haven't been that many of those type of devices deployed into the real world. On the other complete opposite end of the spectrum, you have optimization that I think ought to occur on the scale of months and years. Because as humans, we're continuously adapting, we're aging, and our needs evolve over time. And one big practical limitation for people who use these devices in the real world is that generally, if it's a medical use, insurance won't pay for a new device except every 4 years or so. So if you experience, a physical change in your body, that your experience from 1 month to the next is suddenly different, you may not be able to optimize. So when you ask me about optimization, I can't help but think about the practical barriers. When I first got into the field, I was very excited about this idea that, well, we just strap a robot on the person and we use AI to continuously learn their movement patterns and optimize the trajectory of torques while they walk. And a lot of people are working on this. I think it shows a lot of promise, but we need to solve the challenge of kind of commercially translating these sorts of products, and it's pretty hard.
Yeah, getting started and asking how hard could it be is a bit of a dangerous game in my experience. Okay, Josh, thank you very much. That was also really fascinating. So with that, I'd like to invite our panelists back to join kind of a bigger discussion here. And it looks like we have about 20 minutes left on the clock. And so what I'd like to do is open it up to everybody to ask questions. As you might have questions, please feel free to type them into the chat. Not everybody has to answer every question. We had one. We had a quick one for Hojun. Thanks for presenting very interesting research results related to brain signals in the last several decades. We've had great progress. Any comments on how to increase the real-life applications based on EEG outside research labs? If you can answer that one in like 15 seconds, that would be great.
Rob, you're making my life more difficult than it has to be with that request, but I'll try my best. No, but I think EEG can definitely be an interesting tool that could be used outside of a laboratory in a way where if it can somehow build some type of a closed loop IoT type of system where someone at home can utilize off-the-shelf EEG to record their brain signals. Because brain signals are obviously, I talked about them in the context of Alzheimer's disease, but brain signals that are recorded through EEG have been studied for many other brain conditions, both healthy and diseased. So I think there are definitely different ways to think about how to use EEG outside of a laboratory and maybe kind of integrate that into even some type of wellness programs or other daily activity monitoring programs. I think that could be an interesting area, but I'm not an expert in that particular area, so I won't go any deeper than that. But I hope that answers that question.
Thank you. And yeah, I think that's a really interesting point, like talking about how um, in the future it seems like we're going to have more and more smarter devices. Like we've already started with Fitbits and everything monitoring um, sleep and our exercise habits and that sort of thing. But like it's going to make sense to have more and more sensors, but then you have compliance issues and size issues and then data issues, and then there's a whole host of problems that go along with that too. So thank you for so succinctly summarizing that. So one question for everybody. This one's a little bit broad. So what has you really excited about the future of research? And by that, what I'm seeing is that everybody talked about like slightly different things, but there was a big trend for everybody in essentially like interdisciplinary research, like using multiple algorithms or data types or subdisciplines or that sort of thing. So like, is that maybe an example of a future research trend that might excite you? And what kind of skills do young people need to seek out in order to really effectively engage on that front?
I suppose I can start. My field is sort of ridiculously interdisciplinary, which is fun for me, but often a challenge. It requires so many different skill sets, mindsets, the types of facilities to conduct a successful project, especially when the research is focused towards translation. So that's a challenge, but I've been very excited to see a lot of progress on this front and to see funders pushing towards interdisciplinary collaboration. So some of our, longest and biggest collaborations have occurred because of kind of a special magic that has kind of arose as far as interdisciplinary, crossing interdisciplinary barriers. So yeah, I don't have a recipe yet. I wish I did, because I often get asked, how do you conduct a successful multi-site clinical trial in wearable robotics? I have some of the ingredients, and I see like a sea change coming. But yeah, hopefully that sort of addresses part of the question.
Thank you.
Yeah, I can speak from the molecular biology and genetics research perspective, and I do believe all these individual labs or research units, you need data science skills, and otherwise there's no good experiment plan, no insight for data analysis. Those data-driving approach cannot apply, then we waste all of the resource, and definitely we need fundamental education for students to have a linear algebra and the skill understanding of the machine learning principles. Definitely there's a long way to go to penetrate into the traditional molecular biology field.
Okay.
There's another question.
Yeah, we had another one. So how important is information privacy and data anonymization in your field, and what can we do to address future issues? And I think this is, this is going to be of slightly different importance for people in the academy versus in the commercial sector, but I'm very curious to hear what everyone has to say.
Well, I guess I can maybe start by talking about the information and HIPAA and all those things that we need to consider, especially when you're even in research, when you're doing clinical studies like the ones that I talk about during my presentation. the bar is constantly rising in terms of information security and privacy for patients or subjects who are participating in your study. So I think that requirement will continue to be, continue to be there, and also potentially become even stricter as we go. So I think it's very important to consider that as you're designing a study even in a research setting, but also being in the, I guess, industry now, running my own company that's in infectious diseases and molecular diagnostics. Same thing here with our device design, where again, the FDA is really clamping down on cybersecurity and information security for any medical device that you're developing and trying to commercialize. So I guess long story short, I think that requirement will continue to be stricter and more stringent from where I am. So I think, yeah, it's definitely very important to consider those before you start doing anything in your research, or before you're making your medical devices. So yeah.
So it's not something I deal with in my work directly yet, but thinking about people in the audience that might be thinking about applying AI ML techniques to like issues of human movement, I think it is definitely a barrier. The reason why we can't build accurate simulations today is because there's a dearth of data on how people move. And part of that is that collecting the data is very expensive and slow, and that's why I started the company that I did, because we're helping to rapidly accelerate the data collection. But there are serious issues of data privacy because, imagine if you went into the clinic and there were a system to capture your gait patterns. How does that data enter the medical record, and then how does it get extracted out of the medical record? We're just barely scratching the surface on that. So I'm excited to see how this goes.
Yeah, that's also a really good point. And then the more data we start collecting about individual people, it becomes more and more of an issue to have at least anonymization along with data privacy, because the more data you have about a particular person, especially if they have some kind of rare condition or disease, like the easier it would be to identify them from their data. All right, fascinating. So a question from Thomas. Since at least two of you produce code as research artifacts, toolboxes, etc., do you think the amount of work that goes into that is recognized enough academically? Wouldn't today's scientists be better off writing another paper as opposed to great code? Very good question, Thomas. Thank you for submitting that.
Yeah, so I think I can speak to that a little bit. I agree that open science in this kind of way should be better recognized and has traditionally not been recognized by the academy. A driving motivation for me to produce good code that was robust was to actually save time. And, the way I saw it was saving time will enable me to be more productive in the ways that are rewarded. So I think it's more of an indirect reward versus a direct reward. But I actually would love to see the academy embrace this a lot more.
Yeah, I don't think we have a sufficient or right way to support the great coders. But in the same time, if you, it's not a contradictory to write a good paper, because good paper carries new ideas and insight, and it will go along with the code. And yeah, so I think I have another question from my toolbox.
Yeah, so the question from Vignesh specifically for James was, is your toolbox available in the MATLAB bound formatics toolbox, or is it custom written?
This toolbox is a custom writing, and it does not rely on bioinformatics toolbox. There's some function overlap, but yeah. This is specially designed for single-cell data analysis.
Okay, great.
I'd love to comment on the writing good code question, because we run into this all the time. So we have, I don't know, some number of dozens of different labs developing MATLAB Simulink code using our platform. And I'm shocked sometimes how hard it is to get them to share code with each other. This is despite MATLAB Simulink making it fairly easy, I think. It takes effort to write good code in the first place, and I think in a lot of labs that often doesn't happen. And then it takes additional effort to share and to deal with issues of versioning. I don't have any insights into how to solve this, really. It's something I've been beating my head against the wall for a few years. But it is something that I'd love to work on with MathWorks. So usually the problem is in versioning of our tools, not versioning of the MathWorks tools, because you guys do work so hard to kind of make that transition easy for MATLAB Simulink users. But anything developed on top of MATLAB and Simulink, it's hard for us to keep up.
Yeah, that makes sense, especially as the pace of development seems to really be accelerating itself. Especially also as development techniques and development architectures themselves are changing with continuous integration, for example. So anyways, Josh, glad to have a longer form conversation about that. See how we can better support you. That was not the intention of this whole meeting, but perhaps a fringe benefit.
I couldn't resist. Thank you.
All right. So we have some representation from the commercial side, some representation from more of an academic and research side. So I'm curious, what do you see as the benefits of commercial academic partnerships, and where do you see growth potential there?
I can speak a little to this in microscopy, at least. This is actually something that we've been working on with the adaptive optics work. Several companies have expressed interest in. So I think this is one of the things where you're going to see a lot more implementations of software in these kind of commercial microscopes. And it's actually really exciting because it helps with, again, it's not necessarily something you're rewarded for in the conventional sense, but it helps really get your work actually being used, which I think is kind of the highest compliment you can pay to a tool developer. So that's definitely something that I'm excited about, is having, making something that is actually useful and people want to use it, and it gives some value.
Yeah, that's a really interesting point, because it's then you can also collect requirements directly from an entirely different group of people. So then you're collecting requirements in your own lab. You're also collecting requirements from people who are using the same methodology for commercial applications. So then you wind up having a broader set of recs that you can use to make your software even more robust. And then, of course, increasing the user base, increasing that download count and the amount of stars you have on GitHub. I mean, that's always nice for all of us, right?
Yeah.
Okay. We did have a question about insurance. So what are the positive and negative influences of insurance on research in biomedical sciences? This is not something that has really crossed my radar in the past, at least not directly on the research front, but I'm curious if anyone has run into this or has formulated an opinion.
In my field, insurance has created a lot of the pressure to innovate, like one of the primary reasons for innovation. Needing to get smarter about how you as a manufacturer, you as a clinician gets paid, requires some creative out of the box thinking. But it's also one of the most frustrating barriers to innovation, because as a developer of a new technology, getting it paid for is sort of a chicken or the egg problem. I'm sort of an optimist, so I always try to frame this as like, where do improved patient outcomes and improved financial outcomes best align? And like, I'm going to spend time there.
Yeah.
Because that's where you get your win-win.
Yeah, I imagine it must be difficult working within this well-established behemoth of a system to trying to figure out how to move the needle and change insurance billing as you're developing this new tech. Like that has to be difficult to get by that presumably high initial barrier to entry. Okay. Okay, so one last question that I think maybe we can discuss briefly. What do you see as potential future use cases for AI in life sciences generally, or perhaps directly in your field? And I think we've touched on that a little bit today, but it's a very hot topic and of a lot of interest. So I'm curious to see what has everyone excited about the potential growth on that topic.
Yeah, I definitely believe this. We are at the beginning of this revolution of AI and the impact of the daily life and in the research life. And my students has already changed the way they read the papers. For example, extract the marker genes from a particular given systems. And also, I think there's a long way to develop even more and powerful and different ways to tackle this intellectual linking between different domains of knowledge. If you share the same pattern and in different field, you probably can borrow the algorithm from one field to another. But this kind of work, maybe we can use AI to help us, because there's a human limitation to understand multiple domain of the knowledge. Definitely, we are in the revolution.
AI is definitely becoming increasingly used in microscopy as well. It's sometimes used to, I think it's also well used to enhance, imaging. So by denoising or improving resolution. But there's also an entire subfield of computational microscopes where AI makes the reconstruction of these kind of what they're called programmable microscopes that have really unique properties. It basically makes the computation time more feasible. And so it's enabling a lot of really unique capabilities in that field.
Okay, then thank you very much, everybody, for a very productive discussion. By the way, I'd also like to thank Elvira Sunahaile, who's our principal application engineer, who's really been supporting in the wings and who has helped to evolve this effort into a reality. So I'm going to give her a quick round of applause here. Thanks. So, James, CJ, Hojun, and Josh, thank you very, very much for coming out, sharing a little bit about your amazing work, and for joining us for a little panel discussion. Today, I learned quite a lot, and I really, really appreciate everybody taking the time to share and to answer questions and to levy some of their expertise to enrich all of us a little bit more. So thank you. And with that, I'll call this session to a close. Thanks all for attending.
Thank you.
Thank you. Bye.
Goodbye.